Hello, we are continuing in this video with our Unit 4 of Phylogeny. And in this video, we are going to move into Chapter 3, the evolution of populations. So um, we will focus in this video on Concept 23.1. And the big objective is to describe how genetic variation arises and explain why it is a prerequisite for evolution. I will likely put some more detailed learning objectives up before I post this video, but that's the big idea of where we're going today. So just to review, we, in a previous video, provided a couple of definitions of evolution. First definition is descent with modification, and this was Darwin's big contribution, or one of Darwin's contributions, the idea that organisms that are alive today have descended from previously living organisms, but with modification. They've changed over time from previously living organisms. And then the second definition we gave was that evolution is a change in the genetic composition of a population from generation to generation. And so we're going to be looking more at that level today, a change in the genetic composition. And then we said that evolution can be thought of as a pattern of observations in the natural world, such as observations in the fossil record that suggest that life has evolved. And we looked at various evidence for evolution last time, and that was basically evidence for the pattern that we observe in nature. And then we're going to move into talking more about the process, the mechanisms that cause the pattern of change over time. And we are going to start with genetic variation, which is the raw material for any kind of change in a population. So just to review a couple of key points about evolution, one of them is that populations evolve. Now, natural selection may act on individuals, so certain individuals either survive or don't survive based on their genetics, based on their particular traits. But the change doesn't happen until the next generation. Those individuals that survive have to reproduce, and then the change can happen. So evolution happens at the population level. Individuals don't evolve. So I might work out a ton, I might get really buff, but that is not going to be passed on to the next generation. Those big muscles aren't passed on. So that is not evolution. Evolution doesn't happen to individuals during their lifetime. It happens over generations in populations. Second big idea, which is the foundation of what we're going to talk about in this lecture, is that evolution depends on heritable variation within populations. If that variation doesn't exist, then no evolution is going to happen. So I want to zoom in a little bit and talk about microevolution. So when we talk about a change in the genetics, we're really talking about microevolution, a change in allele frequencies in a population over generations. So that is the definition for microevolution. And there are three main mechanisms that result in change at the population level when we measure it based on changes in allele frequencies. Those three mechanisms are natural selection, which we talked about in our first two lectures, the idea of differential reproductive success based on traits that certain organisms have. And then genetic drift is chance changes to allele frequencies, and we'll talk about those more in a later lecture. And gene flow is when individuals move out of or into a population and they are carrying genes with them. So that's a movement of genes from one population to another. And we will talk about both of these later. It's tricky because these terms have similar names, but they are very different processes. But today we're going to focus on variation. But first, let me just show you this illustration of the three mechanisms that can cause change in allele frequencies over time. The first one is natural selection. So if we look at these hypothetical beetles, some of them blend in better with their background. So these lightest ones blend in best with their background. So they are less visible to predators and they are less likely to get eaten. Therefore, they're more likely to survive and then breed. And so probably the lightest ones and maybe these medium ones will reproduce, and the next generation will have more of the light ones and fewer of the really dark ones that are easy for the predators to eat. So that's natural selection. Genetic drift would be a chance change where maybe most of these individuals get wiped out, but a few survive, and the two that survive happen to be quite dark. So these are the ones that reproduce, and so the next generation is going to be dark. So they didn't survive because they were stronger or better camouflaged or better fit for the environment. It was just by chance that they happened to survive, say, when a flood comes through. The third one is gene flow, and you see these other beetles that have spotted wings flying in and adding their genes to the population, and then that changes the composition, the genetic composition of the population. So these are three different mechanisms that we'll talk about in upcoming videos. But like I said, today we're going to focus on genetic variation. And genetic variation is the raw material that evolution acts on. And so it's really important as a foundation for allowing evolution. So genetic variation we can define as differences amongst individuals in the composition of their genes or other DNA sequences. And this is, again, within a population. Differences amongst individuals within a population in the composition of their genes or other DNA sequences. So we should review a little bit of terminology. A gene, as you'll remember, is a unit of inheritance that consists of a specific DNA sequence. Or we could think of a gene. This is the way we tend to think of it at a molecular level. A gene is a sequence of DNA that codes for a functional product. And usually that functional product is a polypeptide or a protein. So there's a more or less accurate theme in biology that one gene equals one protein. It's not completely technically accurate, but it's a good shorthand. So a gene is a sequence of DNA that codes for a protein. And the proteins control certain traits. So a gene is also a piece of DNA that codes for a particular trait. Then alleles can be different versions of a gene. So a gene could be the code for a particular protein that determines the color of hair. Alleles could be different versions of that gene that control different colors of hair. So blonde, brown, black, whatever. So the gene is simply the hair color, and then the alleles are the actual different versions of that hair color. Phenotypic variation can be distinguished from genetic variation. Genetic variation is differences in the DNA. not all of that shows up as phenotypic variation. So, phenotypic variation would be differences in observable physical traits, like the color of hair, the shape of your nose, also physiological traits like what blood type do you have? So that's phenotypic variation. Phenotypic variation depends on allelic variation, what alleles an individual has, but also can be influenced by the environment. Now, in order for this variation to be passed to offsprings, it has to be heritable. And so, what we're really going to focus on is allelic variation. So, I'm just going to circle that. If I can get a pin to work, we're going to be focused on the allelic variation because that is what is heritable. Genes that are, whoops, I somehow didn't get allelic variation saved. So environmental causes that we've kind of talked about, the environment can, for example, make plants develop differently, but the next generation won't necessarily develop the same way. So it's that if environment causes a change in phenotype, that's not necessarily going to get passed on. But changes in the alleles, oh, it looks like I'm erasing it, but changes in the alleles are heritable. They'll get passed on. Okay. So phenotypic variation can be kind of an either or thing. So when Gregor Mendel was doing his experiments, he experimented with pea plants and he chose variations to look at that were either one way or the other. So purple flowers versus white flowers, yellow pea seeds versus green pea seeds, round seeds versus wrinkled seeds, green versus yellow, inflated pods versus constricted pods. So either or. And if you have an either or trait, then that's typically based on a single gene locus. These kinds of traits are actually pretty rare in individuals, and Mendel kind of lucked out that he found a bunch of traits that were inherited based on a simple inheritance pattern based on just one gene locus. Now, there can be multiple alleles at that locus, but just one gene locus. Most traits are controlled by more than one gene locus, and that results in a range of phenotypes. So you can see with these horses, there's not just black horses and white horses. There are all different shades of horses, and some of them have stripes on their noses, some of them have stars on their foreheads, some of them don't have any coloration on their heads, some of them have stockings that are different color than the rest of them. So lots of different variation in horse hair color, and this is based on multiple genes. So how do we measure genetic variation? There's a couple of ways we can measure genetic variation. We can look at gene variability. So this would be the average percentage of loci that are heterozygous. Now, let's think about the word loci. I mentioned gene locus in the last slide. A locus, you'll remember, is the place on a chromosome where a particular gene is located. So here on these two homologous chromosomes, there is a single locus for the flower color gene. And this particular chromosome that's colored blue has an allele for purple flowers. This particular chromosome that's colored red has an allele for white flowers. So we can look at the percentage of individuals who are heterozygous. This individual that has two different alleles is heterozygous. So their genotype would be AA, big A, little a, for this particular trait. If an individual had two alleles for purple, excuse me, that might be the dominant trait, so they would be big A, big A. If a individual had two alleles for white, they would be homozygous recessive. So the heterozygous is having two different alleles for the same trait. So one way of measuring genetic variation between individuals or among individuals in a population would be the average percentage of loci that are heterozygous within that entire population. Another way of measuring genetic variation is to try to measure nucleotide variability. And so I want to show you a picture of one gene. So this is one gene, and in this figure, we've just divided it in half. So this gene goes all the way from here to here. And in your textbook, it's one long gene. But just to make it fit on the slide easier, we've split it in half. The dark blue regions are the exons that are going to be expressed. And then the more gray regions are the introns that will be snipped out in RNA processing and won't be translated and won't be part of the protein product. All of these little yellow hash marks are places where there are variations amongst individuals. And by the way, this diagram is based on the gene for alcohol dehydrogenase, which is an enzyme that is produced in fruit flies. So researchers analyze the DNA from a dozen or more fruit flies and discovered that there were variations at all of these different sites in this one gene. So there's a lot more nucleotide variation than there is variation at the whole gene level. Now, much of this variation, oh, and by the way, the red arrows are insertion sites where there's been a nucleotide or a few nucleotides added in that spot. All of this variation is neutral. It doesn't cause any change in the expressed protein except for this one substitution that's shown with this black label right here. There's a substitution that resulted in translation of a different amino acid. So there's a lot of nucleotide variability that doesn't necessarily result in phenotypic variability. This one mutation here causes a phenotypic change in the functioning of the enzyme, the alcohol dehydrogenase enzyme. Now, as I said, I just want to emphasize this before we get into what causes variation, that genetic variation is the raw material for evolutionary change. And if there is no genetic variation, then evolution cannot occur. If the whole population is genetically identical, then there's no way for it to evolve by any of the mechanisms that we've talked about. I guess genetic drift, if some related population would contribute some genes that are different, then that could cause a change in that population. But it's not going to evolve by either natural selection or by genetic drift. Okay, so how do populations become different from each other? There are four main mechanisms that can produce genetic variation within a population. So we will look at each of these in turn. First, we'll look at formation of new alleles. The main way that new alleles are formed is by mutation. So many people would say that mutation is the source of the raw material that evolution acts on. So mutation is defined as a change in the nucleotide sequence of an organism's DNA. This can result from several different things. There can be errors in DNA replication. When the DNA is being copied, sometimes there's a mismatch of nucleotides. Usually, the DNA synthesis machinery has ways of correcting those mistakes, but occasionally there is still an error that causes a change. Another thing that can cause a mutation is radiation, such as ultraviolet light. Ultraviolet light can cause breakages in the DNA chain. And again, usually the DNA repair mechanisms can fix that, but sometimes when they fix it, they put in the wrong nucleotide, and so sometimes you can get a mutation. Sometimes these kinds of mutations will end up causing changes like cancer in cells. And then mutagenic chemicals sometimes cause the DNA to form crosslinks between different parts of the strand. Sometimes the chemicals can change the DNA. So these are ways that the DNA can be changed. Most organisms are well adapted to their environment. And we talked about that in one of the last couple of videos, that most organisms are very well adapted. Therefore, if there's a change in the DNA that causes a phenotypical difference, that is quite often harmful because it makes them less well adapted. Occasionally, even if there is a harmful mutation, oftentimes if it is recessive, it will persist due to heterozygote protection. So let's see what I mean by that in this next picture. So an example of heterozygote protection would be with the sickle cell allele. The sickle cell allele is a hemoglobin allele that has a change from a normal hemoglobin DNA. There's just one change where an adenine replaces a thymine. So that is a point mutation. This causes hemoglobin to form fibers in a low oxygen condition that causes the red blood cell to change its shape and take on the shape of a sickle rather than a normal biconcave disc. And these sickled cells are inflexible and they can get clogged in arteries, and this can cause a lot of pain and it can be fatal. But normal red blood cells are much more flexible and they can pass through arteries and veins more easily. So it turns out that individuals who are heterozygous, they have one normal hemoglobin and one mutated hemoglobin. These individuals can live relatively normal lives. They will have some sickling of their red blood cells, but they can survive. And that is because this gene is somewhat recessive, so it is protected. So in an individual that is heterozygous, and again, that individual would be, like, for example, big A, little a. The trait that is expressed is the dominant trait. And so if the harmful allele is the recessive trait, then it can persist in the population because it's covered up or hidden or protected by the dominant allele. It's only if you have two individuals that are heterozygous like this and they reproduce and one of their offspring might have two little a's, then that offspring is going to have a deleterious effect and probably won't survive as well. But in a large population, these can survive for hundreds of generations, even if they are harmful. And even these harmful alleles could provide a variation that may be beneficial later. And again, the sickle cell trait is a good example of this because individuals who have the sickle cell trait in other words, they are heterozygous, they are better able to resist the malaria parasite, and so they're better able to survive malaria. Um, Now, many mutations are neutral. So we talked about how most mutations are harmful. Many mutations are neutral because maybe the point mutations happen in non-coding regions of the DNA. So here's the information flow diagram where DNA is copied onto an pre-RNA transcript. Parts of that transcript, the introns, get snipped out. And so if a mutation happens in the DNA that codes for an intron, then that's likely going to be neutral. It's not going to cause an effect on this processed mRNA, and it's not going to cause effect on the protein. Or a point mutation, I should have said, in an exon that doesn't change the amino acid. Actually, let me just change that while we're here. Let me change this to exon, if I can write. So point mutations in exons, that should be an E if you can't tell, that don't change the amino acids are also neutral. And let me show you how this could happen on this slide. So again, here's the DNA molecule, and a section of it would be a gene, and a gene consists of a section of DNA. It would be a lot more nucleotides than what are shown here, but you'll remember that the code on the DNA is copied onto messenger RNA, and then when it's translated, when the messenger RNA is translated into a protein, every 3 nucleotides codes for 1 amino acid. So this first codon, UGG, codes for tryptophan, and we can find tryptophan up here, and that's the only sequence of 3 nucleotides that codes for tryptophan. So if we had a mutation in this codon, then it would cause a change to the amino acid. But let's look at this glycine GGC, and you can see that there are 4 possible ways of getting a glycine. You can have GGU, GGC, GGA, GGG. So if there's a mutation in this third base of the codon, that is not going to change the amino acid that is produced. So a mutation there, it'll be a nucleotide mutation, but it'll be a neutral variation. It won't cause a change in the amino acid. So many mutations are neutral. And then occasionally, rarely, mutations are beneficial and they can actually give an individual a survival advantage. Now, I should emphasize that only mutations that occur in the germ cells are going to be passed to the offspring. And remember, the germ cells are the diploid cells that divide by meiosis to produce the gametes. If your mutation is in a germ cell, then it can get passed on. If not, if it just happens in your skin cell, it won't get passed on. Or if it happens in a muscle cell or any other somatic cell, it won't get passed on to the offspring. Okay, second major mechanism for causing variation is altering the gene number or altering the gene position. So this is not going to change the actual DNA sequence of a gene, but it might make more or fewer of that gene, or it might move that gene to a different position in the chromosome or onto a different chromosome. So chromosomal changes might delete genes, might disrupt them. If you have a piece of a gene, if you have a piece of DNA that moves, let's say you have 2 genes, and let's say a piece of 2 genes gets snipped out and moved elsewhere, then both of those 2 genes would be disrupted. Maybe I should draw that out. So if you have a gene that goes from here to here, and then some intervening DNA, and then another gene that goes from here to here. So let's say this is gene 1, and this is gene 2. That's a 1. I didn't make a very good one. And this is non-coding DNA in the middle. Um, if a piece of DNA, maybe I'll change my ink color to black. So if this piece of DNA right here gets moved to someplace else in the chromosome, then both gene 1 and gene 2 will be disrupted. Or a chromosome change could rearrange the genes, leave the genes intact, but they could be rearranged. Usually, again, these kinds of changes are harmful. However, sometimes they're neutral. Sometimes if the genes stay intact, they can be on different parts of the chromosome and they can still function as normal, as long as all the transcription factors work. And again, rarely they can be beneficial. Gene duplications is when genes are copied. And again, this is often harmful to have extra DNA, but if it's not harmful, it can end up expanding the genome and providing more variation on which evolution can act. So, how can gene duplications happen? Sometimes there's errors in meiosis. So, during crossing over, if there's a mismatch in where the chromosomes cross over, sometimes there can be an unequal crossing over, and sometimes instead of equal sections of chromosomes being switched between the two homologous chromosomes, sometimes there's a gene missing that gets switched over, and so one chromosome will be missing a gene, and another chromosome will have an extra gene. There can be slippage during DNA replication, where a part of the DNA can be copied twice. And then there are transposable elements, and I'll show you a diagram of this. A transposable element is a piece of DNA that can move to another segment, to another location in the genome. So one way this happens is that a transposon will be copied, and that copy will move to a different location in the genome, and now you'll have two different copies of that same gene. So again, you can have more DNA. These changes can end up expanding the genome and provide more variation that evolution can act on. A third mechanism that increases variation is rapid reproduction. Now, reproduction itself doesn't produce new variation. So it doesn't matter how fast you reproduce, that's not going to cause mutations or anything. However, in populations that reproduce rapidly, they can accumulate mutations rapidly, and they can change in a relatively quick time period. So, for example, bacteria can quickly develop a resistance to antibiotics, at least in a time frame that seems short to us because our reproduction rate is so much slower than the bacteria. And then fourth, the last mechanism is actually the main source of variation in populations. And I think one myth when people are studying evolution is they think that there has to be a mutation before a population can evolve. And usually there's a lot of standing variation within populations, and most of that is due to sexual reproduction. So we talked about this a little bit in our reproduction unit, how much variation there can be caused by sexual reproduction. One source is crossing over, which makes individual chromosomes have different combinations. And then independent assortment means that the chromosomes that come from the dad are not all going to line up and go to the the gametes. So there's a bunch of different combinations of gametes you can have just by independent assortment, and then you have to add on to that all the crossing over within the chromosomes that happens. And then finally, fertilization, there's a myriad of different possible sperms that could fertilize any individual egg. And so there's a huge amount of variation that can be caused there. So all of this variation from mutations, and we'll go back to this slide, all of these sources of genetic variation can produce a lot of variation within a population, and that is the raw material that is available for evolutionary change to happen. Okay, that's enough for this video, and we will continue on in the next video with looking at some mechanisms of evolution.